Hello, welcome to Plant Science Simplified. In today's topic, I'll deal with energetics of respiration. To understand energetics of the respiration, we have to consider overall reaction of the respiration, which is as follows. So overall respiration of reaction will contain 1 mole of glucose, 1 mole of oxygen. When it oxidizes, it produces carbon dioxide, water and energy. So this is the very simplified reaction. So when we balance this particular reaction, as it is a chemical reaction, we have to balance. So we can see that there is 6 carbons we have to balance, and 6 carbons in the reactant side and product side, and we have to balance 12 hydrogens which have been balanced, which are in glucose, and they will be part of water, and 18 oxygens. And when we do this, we can get 38 ATPs. And how to understand further this? Here the first thing is, first we have to understand that how the glucose gets breaking down into 6 carbon dioxides. So let us understand that first. So let us consider here, we know that in respiration there are 3 very distinct stages which we are studying the respiration, namely glycolysis, Krebs cycle and electron transport system or oxidative phosphorylation. So here the glucose which has entered into the glycolysis, it takes different shapes. So being a 6 carbon compound, first it undergoes the glycolysis, and in the 4th stage of glycolysis we know that there is a splitting takes place, or which cleavage reaction takes place, which makes a 6 carbon compound into 3 carbon compound. Later in 5th reaction the isomerization takes place, and 3-phosphoglyceraldehyde will be the molecule which is continuing the glycolysis. Since 2 moles of 3-phosphoglyceraldehyde will be produced here, the further reactions will be carried with these 2 moles of the molecules. This continues the glycolysis stages, and it ends with the formation of pyruvate, which is again 3 carbon molecule compound. The pyruvate, when it enters the aerobic respiration, it first if it enters the aerobic respiration, it moves into the acetyl moves into the cell mitochondrial matrix in the form of acetyl CoA. So here acetyl CoA is a 2 carbon compound. To become a 2 carbon compound, the pyruvate loses the carbon dioxide carbons in the form of carbon dioxide, and it becomes 2 carbon compound, and acetyl CoA is a 2 carbon compound which is ready to undergo further reactions of citric acid cycle. So here the citric acid cycle begins, and we know that the acetyl CoA combines with a 4 carbon compound oxaloacetate and to become citric acid, which is again a 6 carbon compound. This citrate, they undergoes several 2 more reactions, and isocitrate will form alpha ketoglutarate by losing 1 carbon from each molecule. As a total, it will produces 2 carbon dioxides, and it becomes 5 carbon compound. And next immediate step, the alpha ketoglutarate becomes succinyl CoA by releasing 1 more carbon from its molecule in the form of carbon dioxide. So if we balance, if we observe the 6 6 carbons of the glucose have been balanced here in the form of carbon dioxides which are produced. First two carbon dioxides will be produced at the juncture of cytoplasm and mitochondria outer membrane to form acetyl-CoA. Second set of carbon dioxide molecules will be evolved in the during the formation of alpha ketoglutarate, and third set of, that is two molecules of carbons, will be released during the formation of succinyl-CoA. So this is with respect to the carbon. Now when we further study the stages of respiration which are result into the ATP production, that is the third very important component of the product side. So let us look water in the later stage. So stages of respiration result in the ATP production, or ATP here refers to adenosine triphosphate, and adenosine triphosphate can be produced in two methods in respiration. First method is substrate level phosphorylation. There are three reactions which produce a substrate level phosphorylation and ATP. First two reactions are coming under glycolysis, that is the seventh reaction of glycolysis, where 1,3-bisphosphoglycerate becomes 3-phosphoglycerate. While doing so, it produces 2 ATPs. And the 10th reaction, where the phosphoenol pyruvate becomes pyruvate, and it also produces 2 ATPs. And there is one more substrate level phosphorylation which comes in the citric acid cycle, where the succinyl-CoA becomes succinate by producing 2 ATPs. So together they produce 6 ATPs in the form of substrate level phosphorylation. Next, the second type, very important type of ATP production, is oxidative phosphorylation. Where it occurs? Oxidative phosphorylation occurs in electron transport system, which is present in the inner mitochondrial membrane, cristae, the folded membrane of the mitochondria. There the electron transport system is situated, and there it occurs. But from where it gets the molecules? What is the connection between the rest of the process, that is glycolysis and citric acid cycle, to the electron transport system? To conduct the oxidative phosphorylation, it needs sources. So electron proton pumping cofactors are required, and those cofactors will be generated during glycolysis and Krebs cycle. And we have to understand the various reactions, basically 6 reactions which are involved in the formation of oxidative sources, where it forms NADH or FADH. So let us understand one by one. The sixth reaction of the glycolysis, 3-phosphoglyceraldehyde converting into 1,3-bisphosphoglycerate will yield 2 NADH, and the preliminary reaction to the citric acid, that is the pyruvate getting converted into acetyl-CoA, that also give rise to 2 NADH, and the third reaction, that is isocitrate to alpha ketoglutarate, will yield 2 NADH. Then this is the fourth reaction. This is the fifth reaction, alpha ketoglutarate to succinyl-CoA, that also yields 2 NADH. And you can observe that these 3 reactions are there along with the formation of the carbon dioxide also. And this is the reaction, very unique reaction, succinate to fumarate, where instead of NADH, the NADH stands for nicotinamide adenine dinucleotide. H stands for reduced form, plus stands for oxidized form. Here also the FAD stands for flavin adenine dinucleotide reduced form. So here FAD will be liberated. And the last reaction of the citric acid cycle also produces 2 NADH, where the malate gets converted into oxaloacetate. So let us have cumulative results here. So in together out of 6 reactions, 5 reactions we can see the production of 2 NADH in each reactions. Together it produces 10 NADH molecules, and there is one reaction which produces 2 FADH. Sometimes it's also written as FADH2, but here I have used FADH. So FADH molecules. And where it occurs? Ultimately ATP production occurs in electron transport system, as I already explained. Here there are certain variations among the production, exact number of the ATP production per se, because as the scientific investigations are increasing and so many scientists are working on the same aspect, sometimes we'll get different data. And traditionally we have learned that ATP 38 ATPs will be produced, but it need not be true. The calculation of ATP, but here I make an attempt to say how it is done. So the overview of the method to classify, that's what I want to going to explain here. So usually we consider 1 NADH at the end of when the after terminal oxidation, the proton pump will be there, and those protons will go into the chemoosmosis. ATP synthase performs the chemoosmosis and it produces the ATPs. This is how that's what we have studied already in the electron transport system. So according to that, 1 NADH will produce 3 ATPs, but some literature suggests that 1 NADH produce only 2.5 ATPs. That means the protons which are generated in the electron transport system for 1 mole of NADH is capable only to produce 2.5 ATPs. That's what the meaning is. Whereas FADH, we usually traditionally we fix that it has a 2 ATP, but the recent studies tells that it is only 1.5. And one more aspect is there, the NADH which is getting prepared or which is getting synthesized in the glycolysis, which takes place in the cytosol, and ETS system is present in the mitochondrial matrix. So that particular NADH which is getting diffused into the mitochondrial membranes, it may not generate 2.5 or 3 ATPs per molecule, so its efficiency will be as good as FADH. That is also a theory is there. So whatever it is, but let us keep usually up to 25 to if it is 2.5 it produces 25, if it is 3 it produces 30 ATPs from the NADH complex through the NADH complex, and complex 2, that is FADH complex, it can produce 3 to 4 ATPs. So finally, when we get the balance sheet of ATP production, we can observe that through substrate level phosphorylation 6 ATPs gets generated, and through oxidative phosphorylation the production of ATP can range from 28 to 34 ATPs. So together we will get 34 to 40 ATPs when we combine these two. That is not the end. So there is one glitch. So we have studied glycolysis, and in the glycolysis, the reaction number 1 and reaction number 3, we have observed that the phosphorylation takes place. by the addition of phosphate group to the glucose or glucose fructose 6 phosphate from the ATP. So ATP will be utilized in these investment phase reactions. So such two ATPs are getting utilized. So whatever comes, we have to finally subtract two ATPs from the overall yield. So at the end, our results may say that for 1 mole of glucose, the cell produces 32 to 38 ATPs. Hope this particular lecture was useful to you. If you like my lectures, like it, share it, subscribe it. Thank you for listening.